So this is the second prep, and we had just finished talking about this kind of oscillating clock idea, where you have waves of gene expression, so signaling, and then the turn on of genes, and then the turn back off of them in a very rapid 90-minute cycle. And I mentioned that anterior posterior identity is set in some way by the number of waves that it experiences. And so we're going to just talk really briefly. I don't want to go into the huge detail. I would rather you have a general sense of how it works than try to memorize things. But basically, the anterior posterior identity along that axis is set by sort of what we would call combinatorial regulation. So you have a bunch of genes, and for example, gene 1 is turned on in all of these tissues, but then gene 2 is only turned on in a subset of them. So there is a particular sort of anterior identity that is set by having just, this gene expressed. And then further posterior, gene 3 turns on, and now we've got these 3. And so this is another region that it's defined by having genes 1 and 2 turned on, but not the others, and so on. And you can see that across the anterior to posterior axis, having different sets of genes activated, interpreted from how many times those oscillations occurred. If they only occurred once or twice or so on, then you just have the most anterior. If they came through multiple times, a few more, then we have the next set, gene 2 turned on, and so on. So that's the idea. This should remind you in some ways, of the ABC flower model, which is Hock's genes, or the bithorax complex that we talked about in flies. It has a similarity to those, and it turns out that in vertebrates, flies have just one set of the genes. So they've got all the antennapedia complex, which we didn't really talk about because they have very sort of specific kind of fates sort of in the anterior part. We just talked about the bithorax complex. But it turns out that mice and humans, so you, have 4 sets of these genes. They're on different chromosomes. And none of them is what you would call a complete set. They're all missing some of them, so they're not complete duplications. But they are still organized the way the antennapedia complex and bithorax complex are organized in flies, which is at the, transcriptionally, this is the direction these guys are transcribed in. And the ones that are transcribed earlier are more anterior, and then they're more and more posterior as you move along. And so they're basically transcribed kind of in the order that they are expressed, and they all have complex regulatory regions. In the mouse, as I said, there are 4 different complexes, and they're called the A, B, C, and D complexes. Notice that the numbers of the chromosomes don't in any way match up with those letters. And the only one I would like you to remember is this one. I'm going to talk about a particular mutant phenotype. So these work very similar to the ways they work in flies, except with greater complexity and more sort of small detail. But in general terms, you have the most anterior ones. They start to be expressed just at just behind the hindbrain. And then the next batch are expressed a little more posterior, and so when you get this sort of anterior to posterior expression, that matches the color coding that's here. Notice also that the most posterior ones expressed in the tail of the mouse, they're also expressed in the limb buds. So these are the fore and hind limb buds. And towards the very end of the semester, we will talk about some of the specific Hox genes that basically set the forelimb versus hind limb identity. So the expression of these genes, which is a way of interpreting the number of cycles of gene expression that have come through as those somites are developing, basically sets the anterior to posterior identity. Now, in mice and humans as well, if you mutate one of these genes, you often will only see a very subtle defect because they tend to have somewhat overlapping expression patterns and functions. And so, but there are a number of types of scoliosis, which you may have heard, a slight malformation of the spinal cord that causes a curvature. And that those are maybe not always, I don't know if they are always, but they are frequently caused by mutations in Hox genes. I had a friend who studied this in zebrafish, and it works in zebrafish the same way it does in mice and humans and so on. Okay, so here's just some images of the little embryos, and there's a couple of things to notice. So the B1, this is just part of the B complex. It shows you the sort of anterior. There's usually very sharp anterior boundaries, and sometimes not as sharp, more posterior boundaries. And they are, like you can see the somites here, and this is underlying tissue. And they're expressed in both the neural tube and in the mesoderm, the somites, but they're not always in register. So like you can see for the B9, in the two different tissues, you have different expression limits, and so they're not perfectly kind of in sync with each other. Sometimes they're staggered a little bit relative to one another. One thing to notice too is sometimes there are these apparent gaps. So this is B4 and it's expressed sort of, you know, this region here. I think that might be the limb bud. No, it's not. But in any case, we're about a third of the way along the main body. And then there's a bit of a gap. You can see there's some expression, but then it's darker again in the tail bud. Yeah, and so just, you're not meant to memorize these, just to see that, you know, the anterior boundaries of expression is further back with each one of these genes. And this is just one of the complexes. Yeah, and so think about this in connection with this idea, because, you know, the most anterior ones are expressed further ahead, but then if you look at the embryos, there's regions where you have, you know, more and more of these genes are being expressed further posterior. And so that's why this general diagram is a good introduction to the overall picture. Yeah, I think that's everything. And this is the only, there's, as I said, the mutant phenotypes are often really, really subtle, like a vertebrate isn't quite perfectly formed. But this is one of the few that actually has a discernible phenotype, and this is a loss of function. This is Hox C8, so C means the C cluster, and the genes are not, the genes are named as they connect to the fly complex. And so you'll notice, like, this is C8, but there isn't any C1, 2, or 3 in this, or any C7. So it's just a kind of a, it's just the way that they've been named. This one sort of corresponds to this one in the flies. And so it's given that number. And when you have a loss of function, which is a deletion, a complete deletion, and when the mouse is a homozygous for that deletion, the first of the lumbar vertebrae, which is the one right below, so there's no rib attached to this one, but this gets transformed into a more anterior structure. So it's kind of like the loss of function by thorax mutants that we talked about in flies, and the reason is the same. So you still have that kind of repression of the more anterior genes, and so if HOXC8 is missing here, it's not able to repress the more anterior gene that is telling in this case, the somites to form a more posterior structure, or sorry, a more anterior structure, and so therefore this structure starts developing as if it's further ahead than it is, because C8 is supposed to be repressing this gene that's specific to anterior, and when C8 is missing, the more anterior genes are able to express further posterior and then turn this into the wrong segment. Now, a mouse isn't going to die because it has an extra pair of ribs or something like that. So this isn't a lethal mutation, but it just, it's one of the few where there's a really visible and clear and easily seen phenotype. Okay, and you can use this to make a connection to the bithorax complex that we talked about before the first midterm. There are these connections, even though they're such different organisms. Okay, so we've talked about the identity of the somites, the anterior-posterior patterning, the waves of gene expression, and the Hox genes. And so now I want to just talk a little bit about how the somites themselves are patterned. Okay, and the somites, which are called, as you remember, paraxial mesoderm, they are going to form dermatome, myotome, and sclerotome. Sclerotome is going to be cartilage, which is then going to be ossified to form the bones of the vertebrae. The myotome is going to form skeletal muscles, so the muscles all around the rib and the trunk and the body cavity, and so on. So along, not your limbs, that's different mesoderm. And then the dermatome, which is going to actually form the dermis layer below. So the ectoderm that forms the epidermis has to combine with the mesodermal dermis to form the actual skin. Okay, and so this block of mesoderm has to get signals and information so that the different parts of it know kind of where they're supposed to be and where they're supposed to go, because each somite produces, a bunch of different tissues. And so it turns out that the somitic cells are receiving signals from a variety of tissues, from the notochord, from the ventral neural tube, sometimes from the lateral mesoderm. So this is the medial, the middle part, and this is the lateral, away from the midline, and then some from the developing epidermis. And so you can see some changes. So the first thing that happens with that block of tissue is that you see it kind of loosening up closest to the neural tube here, closest to the sort of ventral medial side. And this is the part that's going to form the sclerotome. And so it's going to, it's going to be moving into this region here because it's going to be forming the vertebrae around the neural tube. And then the remainder of it kind of flattens out and widens, and then it forms the derma myotome. The myotome, the cells will start migrating away to form muscle, and then the dermatome is like a disc kind of underlying the skin there. Okay, so this is physically what it looks like, with the sclerotome changing first and just detaching, and then the dermamyotome forming, and then later those separating. And so these are the main signals. So from the notochord comes sonic hedgehog. So it's like hedgehog from flies, but it's called sonic hedgehog after a video game character. And the notochord also signals to the neural tube, and helps to pattern it, which we'll talk about later. But signals from the notochord also induce sonic hedgehog expression on the ventral side of the neural tube. So both of these are ending up sending signals to this part of the somite, which means, those are the signals to activate sclerotome development. The cells start to detach, they start to move around into this general region, which is where they're going to start forming the vertebrae later in development, of course. And the gene that's expressed in those tissues is PAX1. So that's specific for the sclerotome. And then the dorsal neural tube is sending Wnt signals, and so is the epidermis up above the dorsal side of the somite, and this induces the myotome. And the genes that get expressed here, these are the only ones you have to remember, MyoD and MIF5. These are muscle-specific transcription factors. So when these cells start expressing that, those two genes, and they start behaving more like muscle cells, they start migrating to their normal positions and forming muscle there. And then there are lateral signals. They come from the lateral plate mesoderm. So there's additional mesoderm down here away from where the somite is, because the somites are not the only mesoderm in the body. And this lateral mesoderm is sending signals to the dermamyotome cells. And then these are going to, we don't have specific gene expression, but they are going to form these, the limb muscles, and those migrate far down through, but the tongue, so up in the head area, and then muscles of the body wall, right? And so, yeah, there's nothing specific to mention about the derma part of the dermamyotome. So, but these are the signals you need to remember. Okay, and that's a very rapid run through how this process works. And next time we're going to talk about sort of signaling of the neural tube, how the neural tube forms, and then a very special tissue, which is actually some developmental biologists consider the neural crest like a fourth germ layer because it has very unique properties. And yeah, and there's some very interesting color patterns in mammals that we can look at based on defects in the migration of the neural crest cells.